Hi everyone. As Adam mentioned, I'm a research fellow here at the University of Melbourne, and it's a pleasure to present to you today. Um, so, as an overview, what I'm going to be presenting on is on our experience with capture-based sequencing approaches and how I personally think they're going to become as common as culture-based sequencing in the next 10 years or so. I'll walk you through a few examples of our work in this space, and hopefully convince you that they could be useful for your own research or practice. I'm just listing my declarations down the bottom there as well. So just one slide on my background. So I'm based at the University of Melbourne at an institute called the Doherty Institute for Infection and Immunity, which is a joint venture between the University of Melbourne and the Royal Melbourne Hospital. Through this, I have an honorary role at one of our reference laboratories called Vidril, where we receive a number of viral and bacterial and other samples from infectious agents, which are part of our routine service work we analyze and report on, but we also have a research arm where we're trying to establish best practice and constantly improve in that space, which is related to the capture work I'll be presenting on today. So the kind of fundamental point I'd like to make to start is that the genomics, the culture-based microbial sequencing that we're all familiar with, is really reliant on there being a viable organism in your sample to start with. So in the case of bacteria, as shown, if you've got viable bacteria to start with, for the most part, you can plate them, culture them in some form or other, and establish effectively an inexhaustible amount of that microbial agent. So you can come back to it, you can analyze it in different ways, and you can prepare nucleic acids to sequence. What we're finding in a number of cases, though, is that we have samples that are positive for a microbial agent, but no longer viable. So they don't carry a viable organism in there that we can culture and then analyze or sequence through these traditional methods. So we're really limited on the number of samples or the type of samples that we can then analyze and do surveillance on. Just to hammer that point home, we did some work back in 2018 here in Victoria where we prepared a collection of all of the gonorrhea isolates from 1 year in our state. So altogether, a bit over 7,000 gonorrhea notifications for the state of Victoria and Australia. And of those, around about 28% had an associated culture. So the remaining samples were either no longer viable now or were never viable and were just nucleic acid test positive. This is seen as a really great outcome. So a lot of countries have that number below 10%, where less than 10% of samples that are positive have a culture associated with them and enable genomic analyses. So there's a huge gap where things are circulating in our communities, which we aren't able to sequence, aren't able to analyze and say, what are the patterns of transmission or what are the dynamics happening in this setting? So we're working with these clinical samples or patient samples that are very finite. Once you've used them, they're done. You can't go back and generate more, which is where target capture, at least for our workflows, come in really useful. So it's independent of microbial culture. You don't need to have cell culture setups. You don't need to have bacterial culture. You're working directly from these samples without a culture step and enriching your target organisms' nucleic acids from there. What this does is it enables surveillance and genomic epidemiology on far more samples, and in turn provides a more complete view of disease transmission and dynamics. So rather being limited to that 10% to 20% of samples that have a culture associated with it, we're now broadening that out to effectively everything that is nucleic acid test positive, we can do genomic epidemiology and surveillance on. Just one slide on the kind of methodology, how this works in the lab. Our typical workflow is that we would prepare a nucleic acid library from a sample. So you'd generate nucleic acids, you prepare them and generate a library, standard lengths to be about 400 base pairs, but you can tailor that depending on your sequencing chemistry. You would then add in a number of blockers, things that are kind of there to make sure that our capture step works efficiently. And then you're adding in a predefined panel of probes that anneal to target sequences of interest and don't anneal to other background sequences. So in the case of a clinical sample from a human, you might not have any interest in the human genome. And so you might have your panel be a pathogen. And so if there is a pathogen in your human sample, you've got probes that will anneal to that, and then you'll be able to enrich that pathogen out of the background of human genome that was in that sample. Your next steps are then to add streptavidin beads that are going to bind little biotin groups that are on each of these probes and enrich the particular part of your library that you want to sequence and preferentially analyze. So hopefully through this workflow, you can see that you start with a complex library, and at the end of it, you've got a library that's really focused on what you're after, what your kind of either clinical question or research question is. You're asking that a lot more directly from your data. And to maybe oversimplify things, but what that allows you to do is really focus in your sequencing. So in this case, I'm viewing it as reads per million towards your area of interest. If your starting samples have a range of target organism, pathogen, virus loads that might represent very few, barely detectable, up to 1 to 2% of your data set, that would be kind of standard for us. And then post capture, you've shifted that dramatically so that rather than spending a lot of time and effort and reads sequencing things that are in the background, you're now focusing on just your target. So really, really beneficial in terms of efficiency and cost per base. A few more steps on kind of how you might look to optimize capture panels. So in the design stage, you might go through a few iterations where you generate a panel that targets your organism of interest or certain regions of a genome. And then once you've run through a few samples, you can see that certain areas, just based on things like GC content or structures that the nucleic acids are folding into, might enrich a bit better than others. So some areas might be under-sequenced and others might be over-sequenced. And you can then use that to inform, iteratively the panel design. So areas that, might sequence poorly, you can provide more probes that target that area. And sequences that, enrich really well, you might not need as many. And so you can change the stoichiometry or the relative abundance of probes to get something that's optimal and gives you that uniformity that you might want from your panel. So that is a kind of background. Hopefully, you've got kind of a handle on what we're working with in terms of we're still doing next generation sequencing, but we're no longer doing kind of untargeted or shotgun metagenomics. We're focusing on certain sequences of interest. And in the microbial space, what we might be interested in is viruses. And so Twist Bioscience have prepared a panel that is a comprehensive viral research panel and includes every known human virus effectively is represented in some way, as well as a few viruses of potential zoonotic potential. So things that say like bat coronaviruses that might not be in humans yet, but might have the potential to spill over. So altogether, this panel represents more than 1 million individual probes that each target a virus sequence, and allows for some really broad detection potential from a sample. If you expect there's a virus in a human sample, this panel should be able to enrich it and allow you to sequence it to a great deal more depth than you would if you were just doing traditional shotgun metagenomics. One of the really strong points of using a capture-based panel approach is that it's tolerant of sequence diversity. So if you're familiar with things like qPCR, as soon as there's a small amount of sequence diversity, even 1 or 2 SNPs, that might be enough for a primer or a probe sequence to no longer anneal, and you lose your ability to detect that pathogen in a sample. Compared to our probe-based strategy, where they're tolerant of, in this case, 10 to 15% is where things start to change, but up to 10%, you've got a perfect annealing. So things could be within, 10% sequence divergence and still capture perfectly. What that allows you to do is to pull out things from known viruses that are somewhat distantly related. So it might be a new seroid bar, it might be a new subtype of a virus that hasn't been described before, but using a panel-based approach, you are enabled to report that, to describe it, and that can then inform design of qPCR or other strategies down the line for more focused diagnostics. So a panel-based approach can recover diverse sequences and potentially novel viruses. Now some data applying this particular panel to respiratory viruses. This is data from our hands here at the Doherty Institute, and we're applying the panel to a number of respiratory viruses. So we've selected 8 really common respiratory viruses in our setting and applying the comprehensive viral panel to those samples, just highlighting again that this is independent of any culture-based or other, selective enrichment from the sample side, is just using that patient sample and enriching nucleic acids using a panel strategy. So what you can hopefully see is there is a trend where we get more data, more viral data from these higher viral load samples as reflected in cycle thresholds down the bottom. So this axis here goes from lots of virus to barely any, if any, virus. And so we get really good genome coverage, a huge amount of data for these strong samples, and a kind of decreasing trend as we go down. Still able to recover complete genomes from samples that have cycle thresholds in the 35 to 40 range, which is usually considered the limit of detection for qPCR. So we're able to get really close to the kind of gold standard for sensitivity. And this assay is informing a lot more than qPCR might in terms of what genotype samples are, or is a lot more tolerant of things like diagnostic escape, where qPCR can fall down with, like I mentioned, just one or two SNPs. If we stratify that data out by different respiratory viruses, you can see that it performs really well across the board. The main dependency is on the starting viral load and the cycle threshold that represents. Um, this data is now accepted in Lancet Microbe, so should be out early next year if you're interested. And I've put a QR code up there for the kind of homepage for the comprehensive viral research panel from Twist if you're interested. We are now using it really routinely. It's something that I think we'll have on the shelf just in case there's a new pathogen of interest or an outbreak. We used it for our first monkeypox genomes as an example in 2022. So it's a really useful platform to have. And in responding to high priority outbreaks, it gives you a lot more advantage than just qPCR strategies. So I've got a series of kind of hypothetical questions, just trying to think through what the audience might be thinking at this stage and how we can um, get to some of those key points for you. So one of the things that has obviously become really common in the last 3 years now, well, is amplicon sequencing, particularly for SARS-CoV-2, and that became a real mainstay across the world as our main way to genotype and to inform the transmission or strains of interest, variants of interest for SARS-CoV-2. An amplicon sequencing strategy works really well for comparatively small genomes, for things that are really clonal. And so that worked perfectly for SARS-CoV-2, which met those criteria. It doesn't work that well for things that have larger genome sizes. So things like monkeypox, it was kind of the limit there. Monkeypox was around 200,000 bases, and some amplicon schemes were able to achieve genotyping of monkeypox samples, but it was kind of teetering on the edge of would it work, would it not? Also, organisms that have higher sequence variation, things like HIV-1, an amplicon sequencing approach hasn't been achievable. And then things that have mixtures of microbes. So if you have a sample, a complex sample that might be a mixed infection, or you might look at multiple things within it, an amplicon sequencing scheme is typically not appropriate for that, given that some things might overwhelm lower abundance organisms in a sample. So that hopefully gives you some kind of understanding of why a panel-based strategy might be preferable over an amplicon-based scheme. People also, and might be wrong here for this target audience, but have this conception that, oh, can we apply this to any positive sample then? And I just keep this slide up as it is very much, you can apply it to any nucleic acid positive sample. There are things that might be serology positive, but nucleic acid negative, things like treponema pallidum and things like that, where a panel-based approach wouldn't be effective as there's no longer nucleic acid in a patient sample. But anything that is, qPCR positive, nucleic acid test positive, would be suitable for a panel-based strategy. We're also considering capture-based approaches as a frontline diagnostic. So things like syndromic tests that you might be familiar with from companies like BioFire can also be designed for panel-based strategies. So in this case, I'm highlighting some of the potential pathogens or causative agents for pneumonia, and how having a qPCR test or a panel-based test for multiple different pathogens can get really complex really quickly when you're considering all of this different diversity of infectious agents or non-infectious agents in the case of some syndromes. And so if we're able to, multiplex this assay in a capture-based format, it allows us to look at potentially hundreds of pathogens simultaneously from a patient sample, from a syndromic patient sample. So we're looking at this for neurological infections, for things like pneumonia. It's potentially a really useful frontline diagnostic for things that aren't getting picked up through routine testing at the moment. And then lastly, people seem to think that a panel-based approach could only be applied to viruses as they're quite small and amenable to that. But we've had a real interest in higher organisms and bacteria and things that have larger genomes. And so this is a bit of work that we've put together over the last few years on Treponema pallidum, which is a bacterial pathogen, the causative agent of syphilis, has a genome size of about 1 million base pairs, and is non-culturable, at least not routinely in most diagnostic labs. And so we didn't really have a handle at all on the genomic epidemiology of this pathogen. And there was a real resurgence of syphilis in our setting in Australia and Victoria specifically. So we needed to kind of have a handle on what was happening and what was driving this local epidemic. And so we designed with colleagues at the Wellcome Institute and applied a panel targeting the entire genome for Treponema pallidum, and were able to integrate that with publicly available data from elsewhere in the world, showing that our local Treponema pallidum genomes were interspersed with other globally relevant genomes. And we didn't kind of have a local outbreak that was taking off. It was just admixture with the rest of the world. And it's likely that syphilis rates are increasing elsewhere in the world. And it's maybe something outside of the pathogen's genome that's driving this. And this work's published in Lancet Microbe if you're interested. And then lastly, and this might be a bit cheeky, but Treponema pallidum has a relatively small genome for bacteria. What about proper bacteria? And so in the last 6 to 12 months, we've been working on a panel-based approach for Mycobacterium ulcerans, which is a large bacterial genome size. It's 6.4 megabases, and it's the causative agent of a thing called Buruli ulcer. So this is a skin disease that is endemic to Australia as well as a number of places in sub-Saharan Africa and elsewhere. And we didn't have a good handle on what the reservoirs were in our setting. We had some evidence that it could be possums, but also some evidence that mosquitoes could be a transmission vector, and that traditionally water sources are seen as a potential source of infection for Buruli. And so we really wanted to get some genomic epidemiology around that to say, where the human cases of Buruli also coming from, what sources are they closely linked with. And for that, we really needed to have a culture-independent sequencing approach, which a panel would work really well for. And so this worked beautifully. We're able to recover Mycobacterium ulcerans genomes from mosquito samples, from possum samples, and to integrate that with the clinical genomes that we had data for. And all three were, indistinguishable in phylogenetic trees, showing that they're really closely related and they're likely, linked. And this is just showing where the cases come regionally in Melbourne. So, yeah, some really clear evidence that mosquitoes are a vector for Mycobacterium ulcerans in our setting. And this work, led by Tim Stenier and colleagues at the Doherty Institute, is in press at Nature Microbiology now. So just in summary, I realized that was a kind of a whistle-stop tour of some of the work we've been doing in capture-based sequencing approaches, but I personally think they're going to become more and more common in the next 5 to 10 years as some of the drawbacks of traditional culture-based approaches are more and more evident, and we really need to have more comprehensive data sets to inform surveillance and other public health measures related to that. And just highlighting some of the examples in Australia and how these approaches are already informing public health in our setting. So with that, I'll just close and acknowledge a number of colleagues who have driven a lot of this work with me here at the Doherty Institute. And thank you for your time.